So a warm welcome to all ISPIC participants, ISPIC 2021 participants once again, and teams to the 7th webinar of the Smart Protein Innovation Challenge India 2021. And this specific webinar is focused on cell culture media optimization. I think a lot of our participants have been waiting for this webinar for quite some time, and please, this is the opportunity for all our teams and participants to shoot your questions off to our expert speaker today. And before I introduce our expert speaker for today's session, I'm Akshay Bhatt, the Cultivated Meat Science and Technology Specialist with GFI India, and I'll be hosting this super exciting session with Kumud Kushwaha. We also have Nicole Roque as well as Devika, my teammates from GFI India today. So before we start our webinar for today, general ground rules as every time we have a webinar, you as a participant can use Q&A section or the button for questions to the speaker. And also please feel free to use the chat section if you have anything specific to share with the rest of the participants and panelists, but please do not use it for private discussions. And also after the webinar, we'll have 20 minutes of Q&A session where all the questions will be answered, as well as if there are any participants who would like to answer the question live, please press the raise hand button and we will unmute you and promote you as a panelist for asking your question live. So moving to our speaker today, Kumud, a warm welcome to Kumud. Kumud is working with Merck in the capacity of manager, a life science regulatory APAC, as well as advocacy, as well as surveillance, and is building a regulatory intelligence in APAC with a focus on food, beverage, and cultivated meat regulation. Her main role is to dissect the regulatory landscape of India and the ASEAN region and facilitate policy advocacy by building relationships with local authorities and industry associations such as FSSAI, BIS, FICCI, and CII. Apart from policy and regulation, she's also very keen in developing new methods on various cultivated meat technologies, which includes new culture media formulations as well as optimizations and scaffolding as well. So warm welcome to Kumud once again. And before we move to Kumud for her presentation today, I just would like to again thank our sponsors. This challenge would have not been a reality without the generous support from our sponsors. Our title sponsor, Capri Global, our key sponsors, NICE and Alchem Foundation, organizing partner, CII-Co, supporting partners, BIRAC, NIFTM, CCAMP, AICCC, MBI, Madras by Incubators, Sign Enable, and our ecosystem enablers, Blue Horizon, and DSG, Fixie, Brink, Ankur Capital, Axelo Labs, ProVeg, Omnimo, Agni, IMSA Ventures, Big Idea Ventures, and Hardy. So again, I think most of you have seen these slides multiple times, but again, once people cross phase 2, that they're actually building the 5-page proposal, you'll move into phase 3, where you'll have an in-depth exercise of building a 20-25-page proposal, whether irrespective of whether in track 1 and track 2. Track 1 is the participants or teams in track 1 are expected to come up with a solution to a specific problem statement, whereas in track 2, it's totally entrepreneurship, and people who are interested in building their own ideas and setting up their own ventures can start across all verticals of smart proteins, that is cultivated meat fermentation as well as plant-based proteins. And the teams which cross phase 3 and qualify for phase 4 will be given a chance or an opportunity to present in front of a panel of investors for raising their pre-seed or seed fund rounds. So we have a prize pool of a total of INR 20 lakhs for up to 20 teams across track 1, that is innovation, as well as track 2, entrepreneurship. So today's webinar is focused on cell culture media optimization, and one of the key RFPs which we have actually put together is finding innovative solutions for low-cost production of recombinant albumin or an equivalent of albumin, that is for track 1. Whereas in track 2, I think this is a great opportunity for you to, clarify your doubts, your questions about what could be some of the more innovative solutions, approaches in terms of combinations of different growth factors, in combinations of different proteins. And let's say it's probably based out of, let's say, fermentation-derived proteins, or it could be through feedstock optimization, side stream valorization, or it could be through plant-based proteins as well as growth factors too. So let's move on to Kumud. So I'll stop sharing my screen now, and Kumud, I think the floor is open to you for your presentation. Thank you.
Thank you very much, Akshay, and thank you, GFI India, for providing me this a platform to share up the knowledge database, whatever is right now we are working around in Merck Life Science as such. So I will just share my screen for today's presentation. Okay. So, Akshay, a question to you: is the screen visible?
Yes, it is, Kumud.
Okay, thank you. Thank you very much. So I would say, like, when we talk about the this particular special area of cultured meat development, it's quite a very dynamic situation right now, wherein we are evolving and we are evolving together, because the kind of advancement which we are seeing over the last decade is huge. If we consider, like, 10 years down the line, if somebody would have said to us that we would reach in a situation wherein cultured meat would be something called as a reality, it would have been difficult to visualize. But it's a continuous effort which we are growing up. So one thing which I would also like to say, that I am not a very good expert. I would say I'm still learning and learning from the community itself, and that's the reason why I wanted to give back in the terms of the information to this particular wonderful community which has been created by JFI India. And so here I am, and the topic today, which we are talking about, which Akshay has already introduced, is basically focusing on media challenges and opportunities. So just taking you through Merck Life Science for the people who are not well versed with Merck. So Mook is a 350 years old organization started out of from Darmstadt, Germany. And we use the trademark of Merck across the globe except for US and Canada. So US and Canada, we operate as EMD Serino, which is part of healthcare business, Millipore Sigma, which is the life science business, and EMD Electronics, which is the high-tech electronics business which we have. So we are like a global company, and cultured meat is something which we are taking looking it at very seriously angle for the curious people. And the way it is drawing the curiosity out of the scientific community. So we have a Merck culture team actually dedicated, which is based out of Darmstadt, Germany as well. They are working really into this different experiments. And during the course of this presentation, I would be sharing some of the findings, what they have done as well. So now moving ahead, that what are the technologies which we have in the present, and we are trying to scale it up. So basically all the four different categories or the dimensions of cultured meat. So first one is which we have the cell lines as well. So we have some of the meat producer cell lines. We are focusing a lot of work which is going on right now into animal component free media. And courtesy to our biopharmaceutical manufacturing, we have some of the bioreactors and bioprocess designs, as well as we are working on edible scaffold. So Merck actually as such is a very complex organization. So we have different businesses, and all these businesses are coming together, because this cultured meat, when we talk about is a new technology wherein there's lots of application from different businesses, and how we can utilize the existing platform and deliver the best possible solution to it. So moving ahead, setting up the context, that you might have heard, like this particular audience would have been already being aware about why we are talking so much of cultured meat. But this slide is just to set the context of the importance of cultured meat. So when we talk about the cultured meat, the first important piece is the environmental friendliness of the cultured meat, as well as the sustainability which it brings around. So if we talk about the environmental perspective, cultured meat results in a 92% lesser greenhouse emission, 87% less land usage, and 94% less water usage as well. And when we move around to the ethical piece, since there is no slaughtering required, it's again moving towards the ethical direction, wherein the sensitive area of slaughtering is addressed over here. Then when we move about to the world population, which is ever increasing, and if we go by the data of United Nations, it says that by 2050, our world population, which is 8 billion right now, will be going across to 10 billion. And the sub-Saharan African region itself is going to get double digit growth. So in the population. So when we are talking about such a big population, we also need to think about the growing demand which will come up for feeding up that particular kind of a population. Also, during our, like, last time also, during the COVID 2019 situation, we understand, like, what does the protein brings about into the diet, as well as there is lots of increase of functional foods coming around. So similarly, there is also, like, it's a well versed old theory that the protein component, especially in the developing nations, is very minimal. And there is also a growing demand which is happening up for animal protein, specifically into countries like countries which are there in Africa, Asia, as well as South America. So cultured meat could be a best alternative to supply this particular growing demand. When we talk about better health options or better health diets over here, when we talk about culture meat, ideally the culture meat will not use any antibiotic, since the animal is out of the equation. Then when we are talking about pathogens, since it's already cultured meat, wherein the utmost care is being taken up and all the traceability, data traceability is there, the cross contamination piece from the pathogens or the parasite free thing is something which is quite possible with cultured meat. Then apart from that, like, we have the option of customization of like, changing up the nutrient level, or addressing the public health concerns or the consumer preference when we talk about cultured meat. So this particular flow chart is something which actually talks about the different stages of the cultured meat, and this is courtesy to GFI. So here we are talking about from the first stage, which is like, the cell isolation, getting up the stem cells, making up the starter cultures, selecting up the cells which we are going to use it up. And then the phase two, which is the cell proliferation phase, wherein we are just focusing on increasing the biomass of the cell. And the third phase, which sorry, the second phase, which is basically the tissue perforation, wherein we are looking at the final product making up, like, in a form of an organ, or the final product, however we are visualizing it to be, so that it can be taken up as the food. Further focusing upon into the culture meat multidimensional field, there are four main technologies which are very evident. So first is basically the cell line. The second is the cell culture media. The third is the scaffolding, and the fourth is the bioreactor. So when we talk about cell lines are the starting blocks and the foundation of culture meat. From one species, different cell types can be used, such as the muscles or the fat cells, which we had seen in the previous slide. Also, for the cell culture media containing the essential nutrients and growth factors also needed for cells to grow and thrive. When it come to scaffolding, scaffolding is the one which provides the structure to the culture meat. So here we are talking about 3D platforms which create a microenvironment needed for cells to replicate and give a product which is equivalent to, like, the conventional meat products. Bioreactors are the platform wherein we are going to increase the cell mass and make the environment suitable for the growth, and then have our final product. So when we talk about right now, the context of work which is going on in Merck KGA, we are working on the two dimensions of this particular technology. That's one is definitely the cell culture media which we are working on, and scaffolding is the another technology which we are simultaneously working on into our culture meat program. Now, looking up at to the different challenges which are there, especially when we talk about all these four important pillars for the culture meat process. So when we talk about cell lines, one of the important thing or a challenge which we face is basically the cell line characterization, which is right now, which is not very much standardized. And what effect does it has is basically on the quality control. So you cannot maintain the quality unless and until you know that what is the cell line characteristics, or how it is going to behave, like, later on of the stage, and whether it is not going to get deviated from the earlier stages. Also, when the cells divide rapidly, there is a very high probability that the genetic content will not be stable. And there are different technologies there, like, single cell genomics, or the epigenomic sequencing, or transcriptomics, which are there, which can actually help us to identify the heterogeneity of the cell. But the point comes up on the cost effectiveness, like whether this particular quality test or quality control test is going to add burden to our final product. So definitely the cost is high. So by the time it becomes feasibly like, cost effective, what are the options being there? Then the another challenge which is mostly faced is on the culture on the cell lines specifically, selection is basically what is the capacity for the replication of the cell lines or the adherence dependence of primary or the committed cells. Then the another point of this cell lines is also the sensitivity towards the metabolite or the shear pressure, which is there in the later on, or the cell proliferation stage or the tissue generation stage. Then how about maintaining this high proliferation and differential potentials, and also having a robust master and working banks? Because as we are already aware about the fact that having the access to the proprietary cell lines is not possible, and these work banks actually becomes I would say a limitation when we are looking at different cell lines and how we can have them for the initial start of the experiments. So here, the solutions which are quite appearing to be potential would be like, for example, when we are talking about transcriptomics or single cell genomics, the cost which they bring a burden to the end product. How about having up a technology which is species specific, using up species specific antibodies, like a format of ELISA, wherein we can use that for the initial characterization, though it would not be equivalent to what the results or the pattern which could be given up by single cell genomics or transcriptomics, but still it would be a decent enough test to have the characterization in place. Then talking about the changes in the cell lines being brought about by genome editing, and then having the custom tailor-made cell lines. But now one of the important piece which we understand is the customer and the regulatory acceptance for genome editing is very less. So for right now, on this area, there is a big question of usage of genome editing. Then, in order to control the shear pressure or the metabolite sensitivity for the cell lines, there is option of having up selecting the cell lines which are able to tolerate higher pressure as well. The membrane stabilization could be via GRAS additives, like generally recognized as safe additives. And how about having a shorter proliferation stage for increasing of the biomass and the other stuffs? Also, when we are talking about the stem cells, there is lots of potential which has been seen for usage of pluripotent stem cells. And the concept of turnkey cell banking is also originating in this particular area. So moving ahead to culture media optimization, or the challenges which we are facing up in culture media. So one of the important challenges is that usage of bovine fetal serum or bovine fetal sera. So when we are using FBS, this is being long aged being used, and there is a reason why it has not yet been completely stopped from the culture from the medium, because of the fact that apart from the amino acids or the trace elements, it provides the important growth factors, and these growth factors are very much important for the cell to proliferate and then contribute into tissue formation. So here when we are talking about cultured meat, the whole I would say the base or the platform gets dissolved if we are going to take in animal origin products into the culture medium, because this culture medium is somehow or the other is going to be part of the cell, which are going to be made or proliferated and then consumed as food. So the possibility of carryover is very high. So here it becomes very much important that whatever we are using up as a culture medium becomes animal free, animal origin free, and it actually goes to the basic that we are not talking about any component which is of animal origin and over here. Then moving ahead at the bioreactor stage, when the cell proliferation is happening or the tissue proliferation is happening up, perfusion is happening up here. One of the important challenge which we are facing with the media is having a stable and a soluble media, because at this stage what we are seeing is when the cell biomass increases and gets packed up, the media which provides the nutrients of this particular nutrients availability to the inner core mass of the cells, it becomes very difficult. So the media should be such that it should be easily transportable among the cells. Then when we're talking about the media, it needs to have a high carrying capacity for high cell density. Then we should have that flexibility for having the species-specific raw materials being incorporated into different culture medias and being used. One of the important thing is which we are facing is having a food grade material and manufacturing the same, because there are a lot of solutions which is being provided into this particular platform, but the consistency of the product availability, especially in the food grade materials, it's quite difficult. And also one of the important thing which most of you are aware, might be aware about, is about the cost load which is being given up by the cell culture media. The cost of the cell culture media itself is actually bringing up the cost of the final product so high that it becomes a problem actually right now. Also, like, lots of things going on how to reduce down the cost of the media, and thus also increasing of the volumes per batch is something which we would like to optimize. And the best solution would be, the lowest possible medium to be used and the highest possible output to be got to get, so that the cost is ultimately reduced out. But there are possible solutions which we are working, also we are looking up towards it as well. There, like for example, when we are talking about animal origin free or having food grade growth factors, how about, like, there are places wherein we are talking about usage of soya protein hydrolysates or some normal or the common food grade materials, which may be lower cost and can be used after qualification for the cultivated meat culture. How about, using up a difference in protein sequence modification to enhance stability and performance of the culture media? So there have been studies, like, in the past wherein, say for example, change in the amino acids sequencing can actually help up in a better usability by these cells. So, for example, there was a study which was specifically, in the bioprocessing division, wherein they had seen that when L-glutamine was used in dipeptide forms, the different dipeptides which were used, so there was a difference in utilization, and the ammonia which was released, or the waste accumulation which was happening, was able to lower down into that. So this particular possibility can be evaluated. Then there are certain growth factor mimics which can be used for production higher potency. And one of the important thing at the bioreactor stage, when the cell proliferation or the tissue perfusion stage, here the waste which are being generated in the media, how about having, like, metabolite scrubbers or oxygen carriers, so that the nutrient availability and the oxygen availability is enhanced at this stage. So this is also something which is very much can be of a important solution over here. So some of the aspects have been studied, and I would be sharing up at the latest stage of the presentation about some of the solutions being worked around for the cell culture medium in Merck program. Then moving ahead at the proliferation stage in the bioreactors. So here we have the different challenges which we face, ranging from sterility to shear force pressure to the gas exchange and the metabolite buildup, as well as the concentration and the harvesting of these cells being very much important at this stage, so that it can be taken up to the next stage of tissue perfusion. Then how about, having up a smaller scale down model which could be easily, like, build up for the later on for the bulk manufacturing. And one of the things which is right now is that we have lots of disconnected unit operations, and how the integration can happen up. This is something, it's like a key challenge. So some of the solutions which are being looked upon in this area are, like, in order to focus on the sterility and avoid any cross contamination, how about, like, having a fully closed system? Then, in order to remove the metabolite buildups, how about having up a novel mixing and a gas exchange mechanism, or an inline metabolite filtration device? Then, talking about the perfusion or the continual processing for this at the cell proliferation stage, and what are the advantages which can be achieved by this? Then there is also, like, lots of process modeling which has been encouraged. So if everything is developed in the lab, and then we are doing up, it actually becomes a very much, I would say, not a cost effective option. So how about using different models or process modeling points from the data points, and mimic the actual process? which can actually help us to understand without much cost investment into it. Then there are solutions like, for disconnected unit operations, there are plug and play modules and peripherals which can be utilized at this stage. Then moving up to the formation of the tissues or on the scaffoldings, and then using up it, making it as a final product. So I would say the amount of research work which is available in this area of scaffolding is very limited. So right now there is not much of information available, but on a top category, it can be like, scaffolds can be categorized into two important pieces. Like one is the naturally derived one, which are derived from cellulose or chitosan, and then the synthetic ones. So both have their advantages and disadvantages when we talk about them. So for example, the natural ones are something like, wherein the molecular weight variation and polymerization is something which is a problem. Whereas when we talk about synthetic ones, the whole development, it's very good thing to have, because you are making it as per to your customization and requirement. But then the regulatory approvals is a problem, as well as the cost is something which comes up. Then the edible material that supports cell attachment and differentiation is something which is right now a lot of work is going on, and there are lots of challenges which we are facing up. And the final harvesting of the after the tissue perfusion is something which is being challenged at. And here there are different approaches when we talk about in the solution format, like, having up the tissue generated. There are different approaches, like the top down approach or the bottom down approach. So for example, so in the top down approach, generally what happens is, like, there are certain options wherein we can have, like, thin sheets of the cells being created and then integrated to form the food. Or we can we can use, like, micro needles technology or microarray needle technology, wherein we can make those vascular structures or vasculatures which can actually help in passage of the nutrients and oxygen to the core of the cells tissue biomass. And also, like, edible plant and scaffoldings are being also being seen at as some of an option wherein we can make it as a viable option as well. So with this, all these challenges and solutions being addressed through, I would just like to take you through this techno-economic study which was done for cultured meat by different, I would say, different organizations and different publications. So here there is this. In the first column, we can see that, there is this an organization which is called CE Delft, which is more known by most of you maybe. So here, what they are doing is, like, they are trying to evaluate the economical evaluation of the end product of the product, and what would be the estimated cost, and what are the different drivers, and how it can be optimized to get this. So here what they are trying to do is, like, they are trying to replace up, for example, they are looking at what are the major factors which is contributing to the increase in cost of the end product. Say, for example, growth factors, as we had discussed earlier, is one of the things which is a major impactful thing. Then the cost of the equipment is something which needs to be brought down at right now with the current stage. So they have estimated that the best cost should be something where around for the culture meat should be $5.66 per kg. So still, this is something it's different at different stages. So another organization, which is open philanthropy organization, which they have also done a project wherein they identified that, the fed batch process with a low cost plant hydrolysate, when they took it up, they found out the cost could be as low as $21 per kilogram for the production of culture meat. Then they also ensured that they also found out that, we have a targeted facility throughput, which can also help us to achieve the limitations of the metabolic limitations for animal cells as well. When we come to our division, and this is like, biopharma division of Merck, when we started initially identifying the cost and how we can reduce down, there were certain things which we identified that, the single cell suspension is something which can be optimized. Then the stir tank for the bioreactors, actually they drive the higher cost. Removing cost of the culture medium from the whole equation actually does not help us to still meet the conventional meat prices. And the best cost which we could get was approximately $1K dollars which is $1,000 per kilogram. So still we are not able to come to a value which is near to the other two organizations, I would say. And if we identify the potential weaknesses, all these three different approaches, they have identified various weaknesses. For example, the bioprocess based on performance expectation of engineered or immortalized cells is something which needs to be looked upon. Then the facility size for exceed animal cell culture bioprocess currently in operation is something very much limited. Then in the other organization, we have seen that the cell mass is, if we consider in the final weight product, is something it's very low. And there is also a low population doubling in the production reactor because of course of the ammonia accumulation which has been seen. And right now at our end also we are facing the problem because we are using the current biopharma processes, which is used to deliver different kind of a product, whereas we are talking about a food product which needs to be economically effective enough. Right now there is lots of extrapolation which we are trying to do to adjust the cost factor, and also at different stage we are trying to identify scenarios which can be used to fix up this particular issues. If we look at the whole thing itself, the main thing which we are looking at right now is the optimization of the cell culture media to bring down the cost, because this is something which we have identified as one of the important things to be worked upon in order to have that price parity. Now, moving towards the regulatory landscape of the culture media requirements. So you guys might be very much familiar about the different countries and what kind of regulations it's bringing up. So, for example, if we look at the different countries, like say, for example, US and Europe, they are the developed economics. So they are advanced in bringing up the different policies into place for the acceptance of cultured meat. So, but can we say that everything has been addressed? No, because this is like a evolving technology, and right now there is not much. There are pathways being laid down by these developed countries, but still, I would not say they have answers for each and everything. When we move to our country, still, I would say we have not yet started our work also in our country. When we talk about culture and meat, we have started taking up baby steps thanks to the organization like GFI India, wherein there are lots of work which is being started to initiate into this particular field. But still, we have to see the sunrise as such. Then there are countries like Thailand, wherein we are seeing that they are also come up with some of the checklist or some of the documents. But when we talk about, say, Israel and Singapore, these are the pioneering countries, I would say. And we definitely, being in Asia Pac, we look up to Singapore in their policy and documentation, so that what could be adapted for the acceptability of the different cell culture medias, or in fact cell culture meat as well. So just moving around to the US FDA regulations right now. So here I would like to bring out to this particular notice that, as I have already said, that US FDA regulatory agreements actually does not solve all the regulatory challenges. As this one is a novel and evolving technology, actually we require both new raw materials as well as raw materials are being identified, and the raw materials which are being used into different contexts are being evaluated for to be used in food. So here, there is lots of work which needs to be done over here. And then also, when we are talking about cell lines, for example, the cell lines, there have been usage, for example, in cheese production and other stuff. But when we talk about the cell lines over here, it is just not just one set of production. It's it's going to be the last product which we are going to consume. So it changes the whole dynamics. Then identifying a food grade cell culture medium is something which can be safe for human consumption at all levels. Still needs to be found in culture meat, and still the work is going on. I would not say we have come to a 100% solution or anywhere. We are just working in progressing into that particular stage. There are different ways of getting up the approval over there in US FDA for the food grade CCM. Either one, we create one, Since we have not created one, how about like, different food additives being used and recognized for the usage? But identifying single each and every food additives would be not commercially viable, because then each of these food additives needs to have a clinical trial, needs to have a result, and needs to be submitted, and then it will be approved. So again, here, like, there is lots of lacunae which we are seeing. Then every ingredient used in manufacture of cultured meat requires a thorough examination for the risk, hazard identification, so that the final food which we are using up is meeting up all the expectations, and it's not introducing any new hazards or new problems into the food context or the food domain. And this one is actually becoming like, one of the pressing points. But still, I would say there's lots of work which is going on over there as well, which we can actually look upon and use it up. So moving to Singapore Food Agency, or SFA, their regulation is something which, if we look upon, and we also, like, it helps us to, as a cell culture manufacturer, cell culture media manufacturer, we look upon at the different stuffs what they are looking up. So for example, SFA regulations are quite very much clear for cultured meat. They are actually mentioning about the different cell lines which are being used, their characterization, the characterization of the meat products. They are also talking about the information is required during the initial approval process for the cell lines which are being used. The information pertaining to the cell culture media is also required, and also the scaffoldings and different materials being used, and their purity level. So if we just focus on the cell culture media, so what are the things required? So they expect that the composition of the media and the identity and purity of all added substances are known, and it will be submitted. And also it indicates that that the companies need to provide the purity of individual substances in culture media, accompanied with the specifications of what they are complying to. Say, for example, they are also recommending that it needs to comply to FAO or WHO committee requirements like the JECFA, BP, EP, or the FCC requirements as well. Also, clarification on whether the culture media remains in the finished culture meat product, it needs to be provided, or it needs to be, if it is completely removed, then the evidence also needs to be provided. So these are all things which needs to be given along with the other safety assessment um, things as well, like, the hazard identification and other stuff. So generally SFA recommends like, takes up 3 to 6 months for evaluation of the documents of the safety assessments, and then it comes up over there. So from this particular piece, let's move to focusing on the cell culture media and how we are changing our, we are shifting our thought process from being using it to, from the biopharma to the food domain. So here, there are some actually best practices which we can apply for media development and manufacturing. So first, a well-characterized raw material can actually help us in maintaining low lot-to-lot variability. Also, the thing is that I don't think that this is necessarily excludes the things like hydrolysates, because when we are talking about maintaining low variability, hydrolysates are basically like, lots of variability exists, but still we need to minimize it over there. Then obviously, if you know exactly what's in your media, you have a better control and consistency, and it will help, like, the manufacturer will be in a better situation to file up for the approval process. Also, there are lots of critical quality attributes which needs to be met, and the regulatory requirements as this regulatory authorities require them. So basically the end result is to have a toolbox of food grade raw materials and media formulations, which can actually help us into different species specific products also, as well as tailor-made custom culture meat and seafood development as well. So these are the different areas which we are right now working around for having a very good media which is suitable for culture meat production. Now, talking about the different media components, first of all, one of the things which we had seen from the DMEM media, which is the minimal basic media or the essential 8 media. When we talk about the essential 8, everybody thinks that it's just the 8 stuffs which is there. But what we are not aware that, this 8 stuffs is actually nothing but 59 different components. So when we talk about a minimal basic media like DMEM, we are talking about 59 components. So the different components of the media includes from right from the inorganic salts uh, to sugar to amino acids, vitamins, trace elements, lipids, hormones, which are the growth factors, and there are different concentrations which actually helps into the development. So for example, when we actually roughly break down this media components and which are listed over here, we find that there is lots of different components which needs to be optimized. So it's not very easy that we are just talking about 8 different components. This amino acids will have 12 to 13 different requirements into it. So when we optimize each and every component of it. Now when we talk about the different growth factors which are right now at the current situation, we are talking about bovine sequencing, we are talking about novel expression platforms and testing of pilot scale samples, which has been initiated for the growth factors development. Also food grade ingredients are also being evaluated, like what can be different food grade ingredients which can be used to eliminate this animal origin products, and have a economically cost viable product as well. So though this is there are alternative methods which are being devised, like as I have already said, like about plant hydrolysates. Because why it's being selected as and looked as one of the important stuff is because of the bulk micronutrients and the amino acids it carries. Then yeast extract, yeast extract also provides lots of nutrient values, vitamins, trace elements, and peptides, and how these can also be utilized. And then we have feed grade ingredients which are being identified and can be developed for the micronutrient level, which can be used up into this culture medium composition, and so that it can be optimized and being cost effective altogether. So moving down to the different experiments, like which was conducted by Merck Culture Meat Media team. So here, one of the things which we were trying to do was having a serum-free expansion medium, and the cells which we had used was QM7 immortalized quill myoblast. So here, the goal was very simple. We need to had a serum-free suspension, and we need to see the expansion of this particular cell line, and whether it was the media was able to maintain the differential potential for the selected cell lines. So the main study revolved around using a basal media screening and blending studies for the serum reduction. And the suspension adaptation here, we were using the platform optimization for cell adaptation and formulating screens. Whereas the design of experiments, we were identifying that, what are the key growth factors which we could identify and use it for the later one. So when we did this particular experiment, we found out that when we use this chemically defined expansion medium, it was able to support the passages of QM7 cells in single cell culture suspension. Also, the cells were able to retain their differential potential as well. And the next step which we are looking at is to whether this could be scaled up to the bioreactor culture level, and also how we can refine the formulation to aid into better serum-free differentiation as well. Now, moving to the different food grade options which are available, food grade additives which are available, which we can use it up for the development this specifically we had used up in quill myoblast cell culture medium. So here what we had done was we had used food grade lysine, we had used food grade NaCl, glucose, corn syrup at different concentration of 30 gram per liter and 60 gram per liter, all pharma grade materials as well as all food grade materials with 30 gram per liter of corn syrup. So why we select later on we selected the 30 gram per liter, because when we did the individual study between the 60 gram per liter of corn syrup and 30 gram per liter of corn syrup, 60 gram per liter of corn syrup was not giving us good results, I would say, whereas 30 gram per liter corn syrup was still better. So we found out that these particular food grade options could be used up into the cell culture media for replacements. Also, the corn syrup supplemented cell consumed disproportionately less glucose than pharma grade control material when we had used. Then also we also found out that the analytical testing confirmed the corn syrup itself had 10% of glucose into it, and the food grade lysine, NaCl, and dextrose had very similar amino acid composition and trace element profiles to pharma equivalents. So this particular experiment actually held us to the belief that food grade raw materials can be, I would say, a very good potential to be utilized as compared to the pharma grade controls which we are using up. So there is lots of scope of using the food grade raw materials in the cell culture medium. Then in the earlier topics of, in fact, when we were talking about the challenges which are faced in the cell culture medium or the bioreactors, we talk a lot about the metabolite accumulation, or the waste cells getting accumulated, and the nutrient being level being less in the culture medium, which results into the death of the cell lines, or not the proper expansion of the biomass which we were expecting. So here, what we had used, we had used a perfusion culture. So you can see here that, just a second. So here you can see that different particular experiment was set up, wherein we had here the I would say, from starting up from this stir tank bioreactor with a heat coating. So it had a foam trap. The glucose was and the anti-foam along with the base could be fed from the top directly. Then there was a pump. There was cell retention device, a feed being there for the cell lines, and the feed balance, as well as the harvest balance and the harvest over here. So here, by setting up this particular perfusion culture system, we identified that it gave best result on the viable cells. Increase was seen as compared to what we can see in fed batch type of reactors. So again, when we were talking about the initial solutions which could be there for the removal of the metabolites and usage of the cell culture media for a prolonged time, perfusion cultures could be one of the best options which we could identify at the earlier studies, which could be used over here. Then moving ahead to the future plan. So for example, for when we are talking about the future plan, here we are trying to avoid using the dry powder media backbones, actually, which is traditional to the conventional culture media. So what we are trying to do is we are trying to formulate all the individual raw materials in concentrated stocks, and then we can use it as liquid handlers to create a set of pH and osmo match formulations as we want on a fly, and whenever we want. So this can be made to species specific cell lines as well, and actually it can help us to make run more complex DOEs, or design of experiments, and it can have a better fit model and optimize formulation faster. So imagine having up a stock which you can custom create and make it tailor made. So right now, this is what which we are right now working around to create a toolbox of different food grade raw materials, which could be used up in the media for any cell type. A process can be developed and custom made at the different stages. So now talking about some of the collaborations which we have done. So as when we started up this presentation, I had clearly said that here when we are talking about, we are not talking about individual work, we are talking about collaborations. So Mook KGA has also collaborated with an organization and it with the CMC, which is Cultivated Meat Modeling Consortium. And one of the major challenges which we face in the traditional research is the number of experiments that we can conduct in a lab. So the number actually depends on various things. Say, for example, it depends upon the lab budget, like how much budget I have got and how many experiments I can run, and then what is the level of automations which I have. So these can be limiting factors. So how about this particular concept came up? How about using different virtual experiments and just using the data points which we are finding it out at a lab scale level, and then extrapolating and finding it out. So Merck is one of the founding members of this Cultivated Meat Modeling Consortium, which consists of representatives from startups, academias, non-profits, and investors. The main aim of this collaborative forum is basically to leverage virtual experimentation by creating a foundational platform for computer modeling and simulations. And there is also a topic which right now it's working around is on metabolic network construction and simulations, which can actually help us providing up insights into a molecular mechanism of particular organisms, and also reconstruct and use it at a bigger level with the mathematical modelings. So this is something which is worked around with that. Uh, uh, there is also a work which is going on thick tissues. So as the majority of you might be aware about that, when we have the cell perfusion level, or when the block of tissues gets created up, there we actually face a lot of problem when we talk about the nutrient excess, or nutrient excess of the cells present in the core of the tissue, or the oxygen and the minerals availability to over there. So how about uh, what should be the right scaffold to be used? So this particular thing is also as a project is being worked upon in this consortium. So I would say like to say thank you for this your time and your listening. And there will be certain times when when we will open up to questions. I would just like to highlight there will be certain questions which I might not be that much proficient enough to answer that. Uh, so I would actually reach up to this particular Merck culture program team only, culture Meek program team only, to get the answers. And for your future also, like, if you have any targeted questions which you would like to ask, so here is the email ID which can get you connected to the experts in Merck who are working onto the cultured meat medium as well as scaffolding work as well. So I will stop the presentation. Over to you, Akshay.
Yeah, thank you very much, Kumud. I think that was quite an in-depth presentation, a lot of technicalities involved. We have an audience who are most of them who are experts in biotechnology, and I now ask all our participants to come up with the questions which most of you have come up with in some of the previous sessions, which was not related to cell culture media optimization formulations. Uh, so we already have, and once again, please post your questions in the Q&A box or raise your hand so that we can unmute you for asking your question. So there are a few questions for you, Kumud. The first one is, this question is from Shailenza Rani. Can species-specific growth factors, example growth factors from chicken or chicken cell lines, or peptides mimicking species-specific growth factors, can they replace FPS basically in cultivated chicken?
So, very interesting question, Shailendra, I would say. And yes, that's the whole thing which we are like, working around of replacing the FBS. Because as I had told earlier that, there is a dependency, I would say, in the conventional culture medium that we cannot, it's very difficult to replace the FBS because the complexity of FBS itself, the kind of growth factors it has. And right now also still there is lots of work which is going on, which we are trying to replace the FBS with, say for example, the experiments which I had shown you about having food grade materials and then using it up in the culture media to give that particular quantity, or the growth proliferation in the cell lines. But the thing is that still there is lots of work which needs to be done over here. But we are definitely looking, there is a lot of potential there, wherein we are trying to identify different food grade materials which could be used as a growth factors for different cell lines. But I would say, like, the journey has just started over there. So possibly it can completely replace FBS. That's the goal itself.
Yeah, thanks so much for that answer. Just to add a little more context here, I think I don't know whether it would be a good idea to actually use growth factors from chicken cell lines. Basically, I don't know what did the participant actually mean. For example, if you want to isolate growth factors by culturing chicken cells, probably that's a bad idea because of the cost concentration and scale-up concentrations. But if you're looking at, let's say, other alternatives, for example, producing it in microorganisms or plants, or like what Kumud mentioned about protein hydrolysates, which could do some of the job of what these growth factors and other proteins do in terms of cell culture, probably that could be a way to go. But yeah, so moving to the next question. Uh, Yadidya has a question. I believe recently Merck is funding a project on spent media recycling. Please share your insights about the research going in this space if you're comfortable doing that, Kumud.
So to be very frank, there is lots of research projects which is right now going on the cultural media space, as I had already told you about it. So one of the things wherein we had reached to a conclusion was the perfusion stage, wherein we found out that having that continuous fed batch is something which is a good option of about having it up, though it not involve the recycling process. But right now the data has not been made available to us, because that's they are working around. You can also write directly to them as well to ask around it. They will be happy to share about the information if it is available right now.
Yeah, thanks
So
very much for that. Yeah, thanks for that, Kumud. Okay, so there is one more slightly technical question. What are some of the positive and what are some of the most effective protein hydrolysates which have been used so far in terms of research? But if you don't have an answer for this, I don't have right now.
So, for example, I can just talk about not very much confidently, but still some of the research papers right now, the work which is going on in I would say the soya protein hydrolysate is being used a lot by a lot of researchers for using it up over there. But one of the thing is definitely the variability which comes up with it. So that is something which is bringing up the challenge in lots of research articles which I can say around. But definitely, if we talk about the protein hydrolysates, Merck is also working around with some of the soya protein hydrolysates to work around. But still, I would say the challenge remains the same, is on the variability and the optimization. Because the moment we shift to reducing the variability, the cost shoots up. Yeah. So, and also the composition factor, for example, we are using soya protein hydrolysate, the result is coming good, but the seasonal variability happens up with the next vendor or the next supplier, then it goes back. So that I can say share about it.
Yeah, so participants can do just like what I did. I have a few questions. That's the reason why I raised my hand. So you too can raise your hands and ask the question live. Uh, but yeah, Kumud, so I had a few questions which participants previously had asked us during uh, the course of this challenge, basically. So, so in terms of media stability, which is also something which I was also, had a few questions or doubts about. So I think media stability is something which is a very critical consideration, right? So in terms of fermentation, probably people use this, or researchers or companies use high temperature, short time treatment in terms of sterilization of culture media, but that's basically for fermentation. So how do you think that the sterility aspects of culture media for CM is going to play out? Do you have any idea about that?
So not, I would say, to be very frank, not very much open about it like right now, because what I could see is still we are using the bioprocessing platform only. So the short time high intensity one which we are using up, there are certain times when we are especially the filtration, the filtration process which we are using it for the sterilization in order to maintain that, because most of the process over there is the components over there is filterable as well for maintaining of the sterility. So still that is not yet touched upon piece, I would say. And to be very frank, this is something which I need to ask the Merck culture team program as well, whether are they looking at different options, because when we talk about different food grade materials, the soya protein hydrolysate we are talking about corn syrups, then how we are ensuring the sterility, or we are just looking up towards the minimum compliance of having it up to a level wherein it does not causes a cross contamination like that way. So that is still I also need to understand a more lot onto that.
Yeah, thanks. We understand, Kumud. So the next question is, I think you brought about this point about this regulatory kind of a hurdle where in which you need to present data in terms of what components of the cell culture media has been assimilated and what's not. For example, if there is any GM component, make sure that that's not assimilated or taken in by the cells as such. But do you have any idea about existing assimilation data which is open access or even as a publication elsewhere? Which compares basically used culture media versus fresh culture media in terms of what quantities of these, let's say, amino acids, growth factors, again, the proteins do change, and what amount is actually taken up by the cells and where it basically resides in the cell. I mean, it's a little complicated, but yeah.
Yeah, it's like a very interesting thing, and it's like, pondered me to go back and then start doing up the research, some of the points which I've brought around. But if I have a look at the different requirements which is there, they have, though they have written, like, Singapore SFA has very much clearly stated, or the FDA also, to the matter of this fact, they have stated that the what are the changes which is happening up and how much is accumulation which is happening up. So this is something which is right now at only the, I would say, the hazard assessment risk analysis stage only, wherein I could see that, there is still a lot of work, because still I'm not able to find up a clear direction from these agencies. It's basically on the like, for example, SFA, as you are aware, like, most of the things is left on the academic or the university professors to decide, because they have created this particular I would say task force team who actually work around and study around that. And there has been several publications also, like I come across like 2 or 3 different publications which has also been released out onto saying about what are the chemical components changes, what is happening up. So definitely, for sure, one thing is we are looking at, is not looking up only to the changes which has been brought about by the cell, but also the concentration which is brought about by the cell. So one of the important things which we are right now looking up at the smaller contaminants, especially on the chemical contaminants, which could be amplified by this cells when they are taken up, especially in the growth factor domains. When we are talking about media growth factors, there are lots of trace heavy metals as well when we have started using up in the media, and these gets accumulated up in the cell, and then, like, those particular tests is something which is identified in the chemical risk identification as a byproduct which is coming from the cell. So this is something which right now is right now on the focus, but there are certain areas which I would still say, like, unexplored into this area, and then could be looked upon. But still, I would not say there is a clear pathway being given that, what could be the things which you need to do it. Like, for example, food, when we talk about food HACCP, it's quite very clear that, you need to just divide into physical, chemical, and biological, and there it is, boom boom boom, you have the matrices which is there. But this is something which you need to think about, and then, come up with it. So I would say it's like, still I do not have all the answers for it, but these, I would say, concentration by the cells, especially from the growth factors coming up, it's something which is right now being explored in Mook.
Yeah, thanks, Kumud. I mean, also, thanks for highlighting about the heavy metals part. So, I think that is a very related question to that as well, in terms of, for example, if you take, let's say, an assumption, basically, a spontaneously immortalized cell line, which is basically, let's say, cultivated chicken cell line, which can be then taken towards preparing or making cultivated chicken meat product. So, over a period of passages, is there any data which exists as to how some of these, for example, heavy metals could change or would be changing the let's say, the behavior of the cells or the genetic mark of the cell, or let's say the lineage of the cell, basically its signature? In other words, is Merck even thinking about in those lines?
To be very frank, no. Right now, no. We are not working in that direction. We are just right now at a very initial stage, wherein, like, as I have told you, the focus is mostly on the cell culture media, getting the right composition and avoiding the FBS. And the second one is scaffolding. But definitely, for sure, like, we have the food testing business as well in the life science. So they are more focused onto how we can test the initial ppm level of the heavy metals or the contaminants which is coming up, or the residues which are coming up. So we have not gone to that particular stage, very frank.
Got it. Thanks, Kumud. And again, a related question: what are some of the regulatory issues related to spent media? Probably we don't have an answer to that, but we can attempt
No,
answering
because
that.
the thing is that the regulatory issues related to spend media is something right now it's not getting focused right now. We have not started actually, to talk about the food which is devoid of the culture media, or talking about a process how to prove that there is no culture media being carried over. So I think so we will come to that particular stage. And if I see at the, like, in Asia Pac, if I look at the developed regulatory requirements which is there in SFA, which I look around, they are more focused on to the end results of the food products rather than like what are the byproducts or the waste which is being generated out for that matter of fact. So right now, no, I could not see anything which is related to spend media.
Yeah, thanks for that, Kumud. Yeah, I mean, also there was a question about, so for example, you said you are using food grade materials basically to formulate your culture media compositions or formulations. Um, so, has Merck or your team or anyone at Merck basically thought about the raw material supply chain for these components, and what do you think the supply chain would look like in the future? Like, what are some of those considerations you have taken into into
So one of the
account?
thing which Merck focuses on, like two aspects for very much, like before we say, for example, we approve anything product, like which we are going to use it for a longer time, is first definitely about the availability and the variation. Lesser the variation, the better it is. So how we can approve? So there is a total supplier approval process into place, and which is very tasking process. I would say right now we are at a very initial stage when we are talking about evaluation. Like once that evaluation is done, then the supplier approval stage will come, wherein we would be like looking around, like how much is the quantity which we require, how much is the supply being there, whether that particular supplier or that particular product is capable of giving up the results which is long run. So that whole I would say, in food terms, we say the supplier development program. So once you identify for a novel products or something which is very much unique, like then also you need to start working on the technology. So how about working into all these particular technology will come from the lab to a bigger level, I would say. But definitely this is something which is always thought through at the initial stage before the selection happens actually. So it needs to be something which we are using up needs to be um, economically viable, needs to be of a lesser variability, needs to be available across yearly, and the quantity which we require. So and something which is also like later on at this stage, because if it's a new food additive, then it needs lots of other different approvals and lots of trials as well. So how about like so it's a very complex process, I would say, and still I would say it's at a very pretty early stage right now, from whatever information I could get from the culture team right now.
Yeah, thank you for that. Just changing the direction of this question, extrapolating it to let's say, so in terms of the opportunity in India, obviously Merck is also, I mean, you are based out of India and you're working at Merck. So what kind of opportunity are you envisaging in India, for example? Is it like a manufacturing capacity at scale? Where do you think India will go in terms of, you could just speak specifically towards culture media itself, and that itself is a big value chain area, right? So,
So that's a very complex question. If I talk about the context of about my organization, so the kind of I would say there are two aspects to it. One is like what we can offer to the culture meet world, and whether our market is going to be something which would be very much of a potential for this culture meet. So right now, because there is the policy also needs to be set up. There is lots of things which we have to still do. So we have the potential for us to be a very good market, but right now I would not say that we have reached that stage or we have even started in that particular stage. Now talking about the what we can offer to say, for example, there is, of course, there is lots of potential what India could offer as a component, because we have seen in the long run in bioprocess environment also, when it started off, India was identified, India, China, in fact, different other countries were also identified as one of the potential raw material suppliers and other stuff. So probably everything is like, I think so, still in the potential stage. Right now we have not yet evolved to like to comment about it as such.
yeah, and probably since the amount of academic research happening in this area is actually multiplying over the last several years. I think it's been much more in the last 2 3 years, if I'm not wrong. Probably this could be a low hanging opportunity in terms of supplying all these culture media compositions or formulations to academic researchers or research groups who can then probably, validate some of these compositions, right? Yeah. Kumud, are you there?
Yeah, I'm there. I think so. I lost in between. The network connection was not that great at my end.
Okay.
Mhm.
Okay. So these are some of the questions. One last question from me is a very simple question. Why QM7 cells, quail muscle clone, ABCC cell lines for standardization? Any specific reason by using this specific cell line?
Mm, no idea. I need to ask them, the creator of the experiments. So this question I can park it up and then I can ask them up, like, is there any specific of using this particular cell line? Because the data actually originates from the from that particular team, so they only know it.
Got it. I mean,
I'm
we,
not
we
aware
have a
about
few cell lines. Obviously, there is a dearth of cell lines which could be plugged into cultivated meat research and development.
it.
But we do have a few other cell lines which I thought would be a much better validation. But yeah, and that's the reason why I asked this question. That's all. Okay, so one last chance for our participants to raise their hands and ask question. If they have something in their mind, please do not hesitate. Any question is not big or small, so please go ahead and ask a question. We'll wait for a few seconds and then we can close out. Okay, I think we do not have any more questions from our participants today, but thank you, Kumud. Thank you for taking out your time and participating in this ISPIC 2021 Inspiration Webinar Series and giving a wonderful webinar on what Merck is doing and what some of the critical insights as to what could be some of the key considerations while working on cell culture media optimizations, and for teams who are aspiring to start their ventures, and for researchers who are trying to build a proposal in this area. And I'm definitely sure that the attendees love this session. And thank you very much once again. And for the attendees, stay tuned for more webinars which are coming up this week. A few on fermentation and one more webinar on cultivating meat scaffolding. So stay tuned and thank you, everyone. Thank you so much. I think we can close out this session.
Thank you very much.